1. Uncovering the secrets of hearing cell regeneration. Zebrafish genes offer hope for hearing loss. 2. Cancer genes linked to future hearing loss treatments. 1. Uncovering the secrets of hearing cell regeneration. Zebrafish genes offer hope for hearing loss. Imagine living in a world where a simple swim could restore your hearing, while humans face permanent deafness from inner ear damage. That's the shocking reality separating us from species like zebrafish, who effortlessly regenerate critical sensory cells. But here's where it gets groundbreaking. Scientists just uncovered 2 specific genes controlling this regeneration in zebrafish, potentially unlocking future treatments for human hearing loss. And this is the part most people miss. These findings might revolutionize regenerative medicine far beyond just hearing. /in while humans can regenerate cells in blood or gut lining, we tragically cannot regrow the delicate sensory hair cells in our inner ears. Damage from aging or noise exposure leads to irreversible hearing loss or balance disorders. Yet animals like zebrafish, frogs, and chicks perform this regeneration naturally. Researchers at the Stowers Institute for Medical Research identified 2 genes, active and distinct support cell populations, that orchestrate this process in zebrafish. Published in Nature Communications on July 14, 2025, this discovery clarifies how zebrafish maintain stem cell reserves while regenerating hair cells. /in/ using zebrafish transparent during development. Scientists mapped neuromasts, sensory organs resembling tiny garlic bulbs along their bodies. Each contains hair cells akin to human inner ear cells, surrounded by support cells. 2 critical support cell types exist: stem cells at the neuromast's edge and progenitor cells near the center. The team found each cell type relies on a unique cyclin D gene to regulate division. When they disabled the gene in progenitor cells, those cells stopped dividing but still transformed into hair cells, proving cell division and differentiation aren't always linked. Conversely, activating the stem cell gene in progenitors restored their division ability. /in this separation of genetic control is vital. It shows how different cell groups within one organ independently manage growth. For example, progenitor cells lacking their specific gene didn't proliferate but still formed functional hair cells. Meanwhile, stem cells preserve their population like a renewable backup system. As co-author Tatiana Petrowski notes, this precision prevents organ depletion while enabling repair. /in controversial angle. Could we eventually edit human genes to mimic this process? The study confirms cyclin D genes also regulate cell growth in our blood and gut, meaning zebrafish insights might apply broadly. But here's the ethical dilemma. Should we pursue gene editing for regeneration, knowing it might disrupt natural biological boundaries? What's your take? Should science prioritize fixing human limitations this way, or does this risk unforeseen consequences? Share your views below. I'm curious where you stand. 2. Cancer genes linked to future hearing loss treatments. Imagine a world where permanent hearing loss is a thing of the past. What if the very genes linked to cancer could hold the secret to restoring our hearing? This isn't science fiction. Groundbreaking research suggests that understanding how certain genes drive cell division, specifically those associated with cancer, might unlock novel treatments for hearing impairment. But here's where it gets controversial. If these genes are so powerful, how do we harness their regenerative potential without triggering their dark side? Researchers at the Stowers Institute for Medical Research have made a fascinating discovery, pinpointing 2 specific cell division genes that are absolutely essential for the regrowth of sensory hair cells in fish. Their revolutionary findings, meticulously detailed in the prestigious journal Nature Communications. You can find the full article at https://www.nature.com/articles/s/41467-02-5-602-51-0, are not just a scientific curiosity. They illuminate an entirely new and promising pathway for developing regenerative therapies specifically aimed at tackling hearing loss conditions. Now, here's a crucial distinction. While certain animals like fish, birds, and amphibians possess an incredible innate ability to naturally regenerate these delicate sensory hair cells in their inner ears, mammals, yes, that includes us humans, have, over the long course of evolution, lost this remarkable capacity. Dr. Tatiana Petrowski, a principal investigator at the Stowers Institute and the senior author behind this landmark study, articulates this succinctly. Mammals such as ourselves cannot regenerate hair cells in the inner ear. As we age or are subjected to prolonged noise exposure, we inevitably lose our hearing and balance. This stark reality underscores the urgency and importance of finding ways to restore this lost regenerative power. So, if we can't do it naturally, how do we learn to do it artificially? The answer might lie in our evolutionary cousins. By meticulously studying the intricate molecular mechanisms that empower other animals to regenerate these vital cells, scientists could gain invaluable insights and critical clues for the development of effective hearing loss therapies. Following this compelling premise, the Stowers researchers employed a sophisticated technique known as single-cell RNA sequencing. This powerful tool allowed them to precisely identify the genes that actively drive hair cell regeneration within two distinct types of support cells found in the common zebrafish, an organism widely studied for its regenerative capabilities. Dr. Petrowski further elaborates on the foundational principles at play. During normal tissue maintenance and regeneration, cells inherently need to proliferate, that is, divide and multiply, to replace those cells that are naturally dying or being shed. However, this process only works efficiently if there are existing cells that retain the ability to divide and replenish the lost ones. To truly comprehend how proliferation is meticulously regulated, we absolutely need to understand how stem cells and their immediate offspring know when to divide, how many times to divide, and at what precise point to differentiate into specialized cell types. By gaining a deep understanding of how these cells regenerate so effectively in zebrafish, our ultimate hope is to pinpoint exactly why a similar, robust regeneration doesn't occur in mammals and, crucially, to determine whether it might be possible to judiciously encourage and kickstart this regenerative process in humans in the future. And this is the part most people miss. The goal isn't just to mimic, but to understand the underlying why. The study's meticulous investigations led to the identification of two pivotal genes deeply involved in the intricate process of hair cell regeneration in zebrafish. The first, the CCND2A gene, was found to be indispensable for establishing and diligently maintaining a robust pool of stem cells during the developmental stages of the fish. The second, the CCNDX gene, played an equally critical role, specifically enabling progenitor cells, cells that are committed to a certain lineage but aren't yet fully specialized, to divide effectively and then differentiate into fully functional hair cells. This division of labor is fascinating, isn't it? One builds the pool, the other puts them to work. When we deliberately rendered one of these genes non-functional, we observed that only one specific population of cells completely stopped dividing, explains Dr. Petrowski. This remarkable finding unequivocally demonstrates that different groups of cells within a single organ can be controlled and regulated separately. This modular control could prove incredibly valuable, potentially helping scientists better understand and manipulate cell growth in other tissues throughout the body, such as the rapidly renewing lining of the intestine or the constantly regenerating blood cells. This separation of control is a key insight, hinting at targeted therapies. Here's where the connection to a more widely known biological process becomes clear. Both of these newly identified genes encode proteins belonging to the cyclin D protein family. This family is notoriously famous for playing a central and often indispensable role in regulating cell division across a vast array of species, from simple organisms to complex mammals. And here's the thought-provoking twist. In humans, the overexpression or excessive production of proteins within this very same cyclin D family has been undeniably linked to several aggressive forms of cancer, including formidable foes like colorectal cancer and melanomas. Does this make you pause? It should. Could the very mechanism that drives uncontrolled growth in cancer be harnessed for controlled healing? Adding another layer of complexity and potential, the researchers further discovered that while each gene was expressed in a very specific type of cell, both of them functionally performed a remarkably similar role in driving cell division. Intriguingly, experiments demonstrated that simply expressing the CCND2A gene in progenitor cells that were otherwise completely missing the CCNDX gene could miraculously restore cell division and normal cell differentiation. This discovery carries profound implications for future research, especially when we consider human models. Why? Because while the CCND2A gene is wonderfully conserved in humans, meaning we still have a functional version of it, the CCNDX gene has, unfortunately, been lost to us over the course of evolution. This suggests we might have a backup or an alternative path already within us. Given that other genes within the broad cyclin D family are already known to meticulously regulate stem cell proliferation and differentiation in other vital organs, such as the constantly renewing lining of the digestive tract and the protective skin, the researchers confidently stated that their findings could have incredibly important and far-reaching implications for medical research that extend far beyond just hearing loss. Dr. Petrowski concludes, insights gleaned from zebrafish hair cell regeneration could, in time, eventually inform and guide research on a wide spectrum of other organs and tissues, encompassing both those that possess a natural capacity to regenerate and, crucially, those that currently do not. This research sparks a fascinating debate. Are we on the cusp of unlocking our own dormant regenerative potential by carefully manipulating genes that have a dual nature? What are your thoughts on using cancer genes for healing? Do you believe the benefits outweigh the perceived risks, or should caution be the guiding principle? Share your perspective in the comments below.